The overall goal of this procedure is to monitor transcript decay rates in proliferating and quiescent fibroblasts.
This method can help answer key questions in the molecular biology field, such as whether changes in gene expression result from changes in transcript decay rate.
The main advantage of this technique is that it provides information on transcript decay rate, not just transcript abundance.
To begin the protocol, establish proliferating and quiescent cultures of fibroblasts. Proliferating fibroblasts should be trypsinized regularly. Quiescent cultures can be established by allowing plates to reach confluence with regular medium changes. Add ACT-D 150 microliters to pre-warmed medium 10 milliliters for all biological replicate cell culture plates. Add ACT-D at a concentration of 15 micrograms per milliliter of medium. Mix the solution well. Aspirate the old medium and add medium containing ACT-D to the cells. To collect the 0 time point sample, gently aspirate off the ACT-D medium and pipette between 5 and 10 milliliters of pre-warmed PBS onto a 100 millimeter plate, 5 milliliters of solution for a 100 millimeter plate, 2 milliliters for a well of a 6-well plate, and 1 milliliter for a well of a 12-well plate. Gently aspirate the PBS. Then add phenylguanidine isothiocyanate solution directly to the tissue culture plate. Pipette the phenylguanidine isothiocyanate solution cell mixture up and down several